you all for the invitation. I am very pleased to present you our results today. I have a lot of colleagues and friends in CRCT, so it's always a pleasure to talk with you. The topic of my talk today is drilling for oils, the role of tumor-surrounding adipocyte, fueling cancer cells to support tumor progression. As Marie told you, my team is located in IPBS Toulouse, and it's totally devoted to study the role of adipose tissue in cancer progression with some emphasis in obesity. So just I will start to have a more general aspect of our studies to told you that cancer is clearly an underestimated effect of obesity. All the people know that obesity is associated to cardiovascular and metabolic disease, but clearly since 2003 and the first study by Eugenia Calais, it is now recognized that there is a link between cancer and obesity. And in this study, she showed that 15 to 20% of cancer mortality in the USA, where there is more obese than in France, is due to obesity. And it's clearly a major public health issue because, as you can see, there is over 700 million of obese and a large more number of overweight patients in the world. I really insist on that because the link between obesity and cancer is not somehow a general effect, but it's depend of first which type of cancer you look at, and second which cancer type you look at. For example, if you look at cancer occurrence, meaning the increased number of cancer, as you can see here, you have an increase in at least 16 cancer has been shown to be significantly increased by obesity. And as you can see on that slide, which is a report by the International Agency of Cancer Research, you can see that the cancers that are highly increased is esophagus cancer, which is due of a reflux between stomach and esophagus. And you see also uterine cancer, it's also increased. And this is mainly due to the ability of adipose tissue to produce estrogen after menopause. So already at this step, you can see that you have big differences between the type of cancer which is increased, and you can already see that the mechanism involved can be clearly different from one cancer to another. So what is the main interest of my team is to look at the role of obesity in cancer progression. It is now clear that obesity worsens the prognosis of breast cancer. It is clearly established, and also prostate cancer and probably colon cancer. You can see prostate, you don't have any increase in prostate cancer occurrence, but the cancer are more aggressive. In general, they are characterized by the fact that diagnosis for both breast and prostate, there is a higher tumor size and increased local and distance dissemination. But when you correct these differences in tumor aggressiveness at diagnosis, you can see that you have an increased distant dissemination and a resistance to treatment. For example, in this study has been performed in breast cancer. It includes more than 50,000 patients. And the key message of this study is that after 10 years, you have an increase in distant metastasis with a 50% increase in obese patients, which is highly significant, but no modification of local recurrence. The main question is what is the mechanism that may explain the increased aggressiveness of cancer in obesity condition. I don't want to spend a lot of time, but you have to think that not all is biology. I mean, you can have delay in diagnosis. The patient, when they apply to screening strategy, the screening strategy as efficient in lean and obese patients to detect cancer, but obese patients are less proficient to go for screening strategy. You have also a problem in therapy during surgery, radiotherapy, and there have been a big debate about the dose of chemotherapy that you are supposed to use. And just to illustrate what I am saying, it's just to show you what it's mean in terms of surgery, the increased time for performing prostatectomy in lean versus obese patient. And as you can see, this is something which is significant. But as you can imagine, we're not working on that aspect. And we know that even if you correct all these parameters, there is in obese aggressive tumor which may be related to the host. So what changed a lot in obesity is adipose tissue. So before I show you the result of my team, some important notion about adipose tissue. In the body, you have multiple adipose depots. The most known adipose depots are subcutaneous and visceral adipose depot. You have also sex-specific adipose depot, mammary adipose tissue in women, and periprostatic adipose tissue in men. And you have also a very specific adipose tissue, which is bone marrow adipose tissue. Despite the fact that they look similar, and I will illustrate what I am saying at the end of the talk with bone marrow adipose tissue, we know that there is differences between these adipose depot in terms of secretory pattern, in terms of metabolic behavior, and it is very important to consider when you are working with cancer to work with the adipose depot which is close to the cancer you are interested in. As I am saying to you, there is a lot of solid tumor which is close to adipose depot. This is the case for example, with breast cancer, which is absolutely evident, but also with prostate cancer. In the adipose depot, we are working with the cells, which is our main topic of interest, which is adipocyte. Adipocyte has metabolic function and also endocrine function, with more than 400 adipokines identified until today. The metabolic function of adipocyte, I will also comment further on. It's to store and to release fatty acid. The last idea, which is also important, is that obesity is not only associated to an increased accumulation of adipose tissue, but also to the occurrence of a dysfunctional adipose tissue. You have hypertrophic adipocyte, which exhibit metabolic dysfunction. You have a recruitment of inflammatory cells, and especially macrophage, and this tissue may exhibit a sub-inflammatory state. So as I told you, the main topic of our team has been to work on the relationship between adipose tissue and cancer progression, and we have been mostly working in prostate and breast cancer. And our key result was to show that surrounding adipose tissue is able to chemo-attract cancer cells to favor local dissemination, as we have exemplified it in prostate cancer. But what is also very important is that when the tumor infiltrates adipose tissue, there is bidirectional crosstalk that is established between tumor cell and adipose tissue that could involve what we have called metabolic symbiosis, that also involve pro-inflammatory factors, and also involve extracellular matrix protein and related protein, which are able to modulate ECM. And what I am going to talk to you today, it's really the crosstalk between adipocyte and cancer cell at metabolic level. Starting the story, it is our first study that we have been published on that subject, which was 10 days ago. And we have made, I would say, a relatively simple experiment to make, I think, an important discovery. What we have done is that we have co-cultivated cells, tumor cells, with adipocytes. And after 3 to 5 days, we have looked of what happened to adipocyte. And what we found is that if you look in this co-culture system, we have found that adipocyte exhibits a decrease in size and number of lipid droplets. They exhibit dedifferentiation process with a decrease in late adipose marker, and they exhibit what we have called activated phenotype, which means that they overexpress ECM-related molecule and pro-inflammatory cytokine. And we have shown that this cell that we call cancer-associated adipocyte promote tumor progression. And when you look at human tumor, you can see that at the invasive front of human tumor, where the two populations are in close contact, you can observe that the adipocyte exhibit a decrease in size and lipid content. And this phenomenon of cancer-associated adipocyte and their decreased size and lipid content has been now described for all the solid tumor. And to my knowledge, there is no example where an adipose tissue in solid tumor at close proximity to a tumor do not exhibit the cancer-associated adipocyte phenotype. By immunohistochemistry, we were able to show that the adipocyte exhibit a decrease in adiponectin, which is a marker of adipocyte differentiation, and overexpress MMP11 and IL-6. And we were also able to isolate this CAA and to perform PCR comparing normal breast cancer with tumor-associated adipose tissue. And as you can see here, we show that we found an increase in MMP11, PI1, and IL-6, and the level of IL-6 was correlated to the tumor size and lymph node involvement. So the first question I want to raise here is what made an adipocyte a cancer-associated adipocyte? It is probably a very complex process. We have shown in our team that it partially related to dedifferentiation process due to reactivation of the Wnt beta-catenin pathway. Some people have suggested that CAA may also arise from a transdifferentiation into brown adipocyte. We can talk about that. I am not completely convinced by the paper, but what is really important is that you have a tumor-induced lipolysis in adipocyte. For those who are not familiar with this process, you know that it is a physiological function of adipocyte to store and liberate free fatty acid upon energy-demanding condition, caloric restriction, or effort. And this lipolysis is regulated by adrenergic receptor, and you will, by successive step, induce the liberation of free fatty acid and glycerol. So there is a clear, and I will show you example, increased lipolysis of adipocyte induced by cancer cell. The first mystery is what is the tumor arising, what is the signal arising from tumor that induce lipolysis in adipocyte. Some team have suggested that catecholamine, which activate adrenergic receptor, may be involved in this effect. We never find any effect in our teams. Pro-inflammatory cytokine, and latest results suggest that a small peptide, which is related to calcitonin-related peptide, adrenomedulin, might be involved in this effect. So adipocyte will liberate free fatty acid, maybe other lipid, and just to know that we have shown in the team that free fatty acid may be involved in extracellular vesicle, but these vesicles are not in our work liberated upon cancer induction, so I will not talk of this part of our work, which has been mainly led by Laurent. So cancer cells are able to induce lipolysis in adipocyte, and it may be considered such as a parasitism. They use a physiological pathway to probably have free fatty acid and maybe increase their nutrient and energy. The question is that, do we have an accumulation of lipids in cancer cell co-cultivated with adipocyte? The answer is yes, but for the moment, the main experiment that we have performed is based on the experimental design, which is the fact that when a tumor or whatever cells have a large amount of free fatty acids that are present within the cell, they will re-esterify the free fatty acid as triglyceride, which will be accumulated within lipid droplet. So what we look at is the accumulation of free fatty acid or neutral lipids within a lipid droplet. We don't really see the flux of free fatty acid, we really see their accumulation. And you can see when you co-cultivate cell with adipocyte, you can see that they accumulate lipid. And for those who are familiar with, for example, in breast cancer, you can see that from epithelial-like cell, you have EMT with increase in membrane protrusion. To really verify that it was really a transfer of lipid and not an increased lipid synthesis in cancer cell, we label during differentiation of pre-adipocyte them with label palmitate. And as you can see on that image, we observe a decrease in palmitate in adipocyte and an increase in palmitate in tumor cell, showing that there is effectively a transfer of lipid between the two populations. And as I told you before, you have an induction of lipolysis. Here it is the two cancer cell that we use. It is breast cancer cell. And as you can see here, when we use normal mammary epithelial cell, we didn't induce adipocytes. It was clearly similar as in control. So the next question, you liberate free fatty acid, they accumulate free fatty acid within the lipid droplet. What is the consequence? Is there an effect on tumor progression? And again, I think it is very important. It is my, I don't know, what I really, for me, it's really important. Look at different adipose depot, look at different type of cancer, and be careful that it is totally impossible to make general assessment, general conclusion. When you look at what has been done, there is lipid transfer from adipocyte into cancer cell. They stimulate tumor progression, but clearly the mechanism involved depend of the tumor type. The more simple or the most common idea, which is also important, is that the free fatty acids that are transferred into cancer cell may contribute to increase fatty acid beta oxidation, and this increase in fatty acid beta oxidation increase ATP, and you know that FAO is important for cancer cell in terms of tumor progression, drug resistance, and survival in stress condition. This pathway has been demonstrated in melanoma and ovarian cancer, I will say with limited experimental evidence. There is two other aspects that I will detail further on. It's one that we have deciphered in breast cancer, which also be shown in colon cancer, and another one that we have shown in prostate cancer. So in breast cancer, what we have observed is that, as you can see here, we have an increase in fatty acid oxidation in a triple negative breast cancer and in ER positive breast cancer, which has quite a high level of fatty acid beta oxidation, because here is a muscle control cell line. So we have an increase in fatty acid beta oxidation. And as you can see, when we use an inhibitor of fatty acid beta oxidation, we observe an increase in lipid droplets, meaning that there is a use of fatty acid which are transferred into cancer cell. And when you inhibit their use, you exhibit an increase in lipid droplet accumulation at atomoxir dose that are quite low. But surprisingly, when we look at the metabolic condition of this cell, what we found a decrease in cellular ATP. We found an increase in extracellular acidification rate in response to glucose. And we found that there was a decreased oxygen consumption rates in co-cultivated cells with an increased proton leak. So all these results suggest that fatty acid, the respiration, was uncoupled to ATP production. And in fact, we found an increase in UCP2 in this cell and a decrease in membrane potential, which were counterbalanced by a decrease in the complex expression of the complex 5 of mitochondria and the overexpression of an inhibitor of complex 5, which is called EF1. So due to the fact that there was a decrease in ATP in these cells, we found an activation of AMPK, and which will in turn inhibit ACC by its phosphorylation. So really there is something like a vicious circle, meaning that you have an increase in FAO, which is uncoupled to ATP production. The decrease in ATP will lead to activation of AMPK, which will in turn favor fatty acid beta oxidation. So the question is, we have FAO uncoupled to ATP production. Does it do something in tumor progression? Because you can see, okay, you have a decreased ATP production, increased glycolysis. Maybe it's glycolysis which is responsible for the increase in tumor progression. But when we co-cultivate cells in the presence of atomoxir, or we perform a decreased expression by genetic approach of CPT1A, which is a transporter of lipids within the mitochondria, what we see is the following. We found that it was able to decrease the pro-invasive effect of adipocyte in vitro. Co-cultivated cells exhibit increased invasive capacity, and it is inhibited by atomoxir. When we inject these educated adipose tumor cells in the tail vein of mice, in the presence of atomoxir and CPT1A inhibition of expression, there was a decrease in metastasis in vivo. And something that for me is probably very important is the fact that we consistently observe a decrease in EMT when the cells were treated with FAO inhibition. And even you can see in control cell, inhibiting FAO clearly reinforce the cell-cell contact in this breast cancer cell line. So to sum up, and what is our idea? We have a transfer in breast cancer of free fatty acid that are used for fatty acid beta oxidation, but this fatty acid beta oxidation is uncoupled to ATP production, but is involved in tumor progression. And our idea is that maybe the accumulation of acetyl-CoA may contribute to the occurrence of epigenetic change, and more particularly to the control of the epithelial to mesenchymal transition program in breast cancer cells. So this paper is, I would like to say, more a proof of concept study in breast cancer, and there is several questions that need to be addressed. As I told you before, each fat depot possesses secretory and metabolic specificity, and the result I have shown you here are obtained with co-culture with murine pre-adipocyte cells. So it is mandatory to perform this experiment with mammary adipocyte because the result can be totally different. So we have now in the lab set up the experimental model to cultivate mammary adipocyte in 3D obtained from mammary adipose tissue distant to the tumors. So this is a very important question that we want to address. Does we find the same result with mammary adipocyte? Another very important question is what happened in obesity condition, and we are also able now to cultivate obese adipocyte, which is quite tricky, and we want to ask the question, is metabolic symbiosis key to the aggressiveness of breast cancer in obesity condition? And one very important question is what's happened in vivo in human tumor, because we have written with Camille from my team recent review about that, and we were very surprised to observe that there is almost no proof that this transfer exists in vivo. We have done some experiments by labeling breast tumor with body pie, and as you can see, we have observed some accumulation of lipids again at the tumor invasive front where the two populations are in contact. But what really we want to do is to use mass spectroscopy imaging and to compare the lipid that are accumulated at the tumor invasive front with the lipid accumulated in the tumor center. And you have some image that have been performed by Landry Blanc, which is currently in Bordeaux. And we clearly hope that soon Landry, which is candidate at the CNRS position, will join our team to perform all these aspects of transfer of lipid in vivo, in murine model, and in human. So that really, I think in breast cancer, it is fascinating, but we have a lot of questions to answer to. So I don't know if I have time. I will show you some results in prostate cancer, maybe escaping some results. We have performed similar experiment in prostate cancer. In prostate cancer, what we found, again, it is simple. We have an increase in induced lipolysis activity. We have an increased accumulation of lipids in co-cultivated cells and a decrease in lipids content in adipocyte, increase invasiveness, and again, increase metastasis in vivo using vein tail assay. And what we further show in prostate cancer is that this effect is increased by obesity, by using human periprostatic adipose tissue from lean and obese patients. We found increased lipid accumulation in tumor cell, and this effect of invasiveness in vivo was more pronounced. But which was quite surprising in prostate cancer is that we didn't find any effect of fatty acid beta oxidation inhibition on the tumor invasiveness. I will go quite quickly on that aspect, but what we have found in prostate cancer, we thought that maybe the co-culture may increase ROS production because, you know, reactive oxygen species is something very important for prostate cancer. And what we found is that we have an increase in ROS production in prostate cancer, and this was inhibited by N-acetylcysteine, which also inhibit the pro-invasiveness of adipocytes. But when we use a specific inhibitor of pathways that produce ROS, we found that only DPI, which is an inhibitor of NADPH oxidase, was able to reduce ROS production and reduce the effect of adipocyte and invasiveness. And the only NOx, there is 5 NOx, which was expressed and increased in co-cultivated cells, was a NOx 5. So to make the link between reactive oxygen species production and invasiveness, we have observed that there was an increase in several metalloproteases, MMP9, MMP14. We sought to investigate the role of IF1, which is the hypoxia inducible factor, but which is also regulated by reactive oxygen species. And you can see that we found that we have an increase in IF1 expression and 2 of its targets. And when we use either DPI or IF1 siRNA, we downregulate IF1 expression and we downregulate MMP14 expression. And we also downregulate the pro-invasive effect of adipocyte. We have a signaling pathway which is present when the cells are co-cultivated with adipocyte and which is increased by obesity. We have an increased transfer of lipid. We have an increased NOx5 expression, and we have an increased invasiveness. We have an increased IF1 expression, and inhibiting this pathway reduce the pro-invasive effect of adipocyte. And more importantly, we found that this pathway exists in human tumors with the help of pathologists from EUCT. And again, as I told you, the crosstalk occurs when the 2 populations are in contact. And we were able to show that we have an increase in NOx5 and MMP40 expression when the tumor cells are located close to adipocyte. And this overexpression was amplified in obesity condition. So our idea in prostate cancer is that free fatty acid may regulate the expression of some gene. We found an increase in NOx5 expression that activate a signaling pathway that is implicated in tumor invasiveness, but maybe other pathways are also involved. As you see, and again, I want really to insist on that. It's not, a lot of people have a very simple minded way to see the crosstalk with adipocyte by saying, okay, it is a tremendous reservoir of lipids close to tumor cell. It will deliver lipid to tumor cell, which is true. And the increase of lipid delivery will be like in physiology for energy demanding tissue, where you will have an increase in fatty acid beta oxidation, an increased ATP, and this increased ATP will contribute to tumor progression. It might exist, but there is also possibility that other pathways are involved, uncoupled, FAO uncoupled from ATP production, and or effect on tumor progression not coupled at all to fatty acid beta oxidation. And we really have to work on that with specific human adipose depot and to really study what's happened in vivo, which is really a missing box in this aspect. So to finish with, I want to talk to you about bone marrow adipocyte, because I know that a lot of people in your institute are interested with hematological malignancy and bone marrow adipocyte. I know that Camille from my team will soon be doing a seminar in Restore, so I will just give you a rapid overview. So we were interested in bone marrow adipocyte because This is a way where you can have metastasis, and we know that bone is rich in bone marrow adipocyte. I don't have time to detail it, and Camille will show you the literature, but the first point, which is very important that you need to keep in mind, is that there is no clear evidence that high proximity to hematological malignancy or bone marrow metastasis, that there is a decrease in bone marrow adipocyte size. So what was really completely clear at the primary site of a solid tumor, it's absolutely no clear for bone marrow lesion. So when we start this project, which was developed by Camille Yatané, the first thing that was really striking for us is that bone marrow adipocyte, it's a very strange adipose depot, because in obesity, you have an increase in the size and accumulation of bone marrow adipocyte, but in opposition to all the other adipose depot, there is no decrease in bone marrow adipocyte, in caloric restriction, and even in anorexia nervosa, which is the extreme of caloric restriction, you have an increase in bone marrow adipocyte, which will lead to a lot of hematologic defect. So the question is that why this adipose depot behave in a such strange way? And when we look at the literature, we didn't find any characterization of human bone marrow adipocyte, and we found only in vitro study. So we decided to set up a collaboration with Nicolas Reyna and to have bone marrow adipocyte coming from hip surgery. So during the surgery, he will aspirate bone marrow adipose tissue, and we obtain pair adipose tissue from subcutaneous and from bone marrow adipose tissue. And if you look like, it looks the same way. When you look at purified adipocyte, what can you see? You see a round cell with a unique lipid droplets, subcutaneous bone marrow adipocyte, which absolutely look the same way. But when we perform a proteomic analysis in collaboration with the IPBS proteomic platform, what we find is that there was no clear difference in adipokine expression, meaning the secretion of leptin, adiponectin, and so on. But when we select the protein involved in metabolism, as you can see on this PCA, we can find striking differences between bone marrow adipocyte and subcutaneous adipose tissue. What was clearly different and clearly differentially regulated between the two depots, the first key result was that there was a decrease in the lipolysis in bone marrow adipocyte, with a very strong decrease of the last enzyme involved in adipocyte, which is mono-MAGL, I would say. And we found also an increase in cholesterol hydrolysis with increased expression in enzyme such as lipase, which is involved in cholesterol hydrolysis. And when we perform unsupervised lipidomic analysis, we found that there was an increase in monoacid glycerol in bone marrow adipocyte as compared to a subcutaneous adipose tissue. You can see here on that slide. And we also found an increase in cholesterol, free cholesterol level in bone marrow adipocyte in comparison to subcutaneous adipocyte. And according to what is observed in vivo, because as I told you, it is very important to underline that these adipose depots do not respond to caloric restriction, we observe that bone marrow adipocyte in human do not express the last enzyme involved in lipolysis, which is MAGL, which could really explain the Mg accumulation in bone marrow adipocytes. And when we look at lipolytic activity induced either by the stimulation of beta-adrenergic receptor or the stimulation of CAMP, as you can see here, we didn't find any lipolytic activity in bone marrow adipocyte, which is in orange, but this pathway was induced in subcutaneous adipocyte. So our key message is that be careful, the bone marrow adipocyte are totally devoid of lipolytic activity, and all the pathway that we have described in solid tumors is not functional for the bone metastasis, which is another problem for what you can see in the literature, is that a lot of people will use in vitro models. For example, the model we currently use, which is F44-3A, which is a pre-adipocyte murine cell lines that you can differentiate in adipocyte in vitro, but also bone marrow mesenchymal stem cells that you can also differentiate in adipocyte in vitro. And we use this model with a collaboration with Gilles Corr that help and provide us the model. So you have adipocyte, they look like adipocyte, and you can see that you have a lot of multiple adipose droplets. And if you look at the lipolytic activity of this model, you found that they are totally able to induce lipolysis. So the problem is that all the paper that has been performed with this model in co-culture should be interpreted with caution, because you really are not working with a model which is representative of the real bone marrow adipocyte. So to conclude with this part, we have shown that bone marrow adipocytes are devoid of lipolytic activity because they do not express MAGL, and they have a cholesterol-oriented metabolism. We have recently published this paper last year, and we think that it may exist a metabolic crosstalk, but this metabolic crosstalk will be different from what we observe at a primary site and may involve cholesterol and cholesterol metabolite. So we are currently studying this aspect, which is a project that has been funded by INCA last year. And we have now set up a co-culture model system. And as you can see, tumor cells accumulate neutral lipids. So there is a metabolic symbiosis, which nature will need to be determined. And this will be a CAMI project. So finally, I would like to thank all the people from my team. It is the current composition of the team. I would like to thank the proteomic group, my friend and colleague Philippe Vallée. I know that he is no longer I2MC, but I keep the slide. We have been working together for a long time. And also I really would like to thank the clinical teams, Mathieu Roumiguier for the prostate, Charlotte Weiss for the breast, Nicolas Reyna for the bone marrow adipocyte. And as you can see on the composition of my team, I am happy that Charlotte and Mathieu are also working in the lab, and it's bringing a lot for our study.